So how is your PhD going on, madam?
So it's going great.
Great.
Actually, I'm working on fungal infection, Candida.
Candida. So
Yeah.
Candida, what, madam? What topic you have chosen?
So I'm doing on multi-drug resistant Candida species.
Acha.
For 50 isolates, I'm gonna do isolation part and several assay, and then I'm going to narrow it down from broad spectrum. So I'll select one or two which is highly resistant,
Mm-hmm.
and for that I'll see the
So right now, how many drugs are available for Candida, madam?
two plants I have selected, which will, yes, sir.
Hello. Right now, how many drugs are available for Candida, madam?
Hello.
Ah, audible, madam?
Very common drugs are only this ketoconazole, fluconazole.
That one it's common.
Sorry?
It is common actually.
Yes, sir.
Other than that, any
Yes,
other
it is very common.
mm, antifungal.
Basic
Mm,
this only. So what I am doing, I am selecting two plants which will show antifungal activity. I have in
mhm. Very nice.
studies on this my
Voice is not clear, I think, madam. Yours.
cell test my highly drug. Uh, yeah, I guess some network issue, sir. Is it
Yeah.
audible now?
Mhm. Yeah. Now it is audible. So shall we proceed, madam?
Yes, sir. We'll continue.
Yeah. Today we are going
I'll
to
start.
discuss about antigen, antibody. Okay.
Yes, sir. Antigen, antibody, and hypersensitivity. It is a continuation
Hypersensitivity,
part of immunology only.
immunology. Okay, madam. Please
Yes.
proceed.
All right, sir. So, hello, everyone.
Voice is not clear, madam. Hello.
Foundation, antibodies, and hypertension. How our immune system sometimes these responses can be exaggerated or harmful. So, antigens are molecules that trigger an immune response, while antibodies are proteins made by our immune system to neutralize or destroy these antigens. Hypersensitivity refers to situations where the immune response is excessive or misdirected, which can lead to allergies and other immune related conditions. So, understanding these three concepts is crucial in diagnosing and treating immune system disorders, infections, and allergic reactions. So, let's explore why these procedures are vital and their mechanisms in detail today. So, introduction to antigen antibody interaction. So, antigen antibody interaction is a biochemical reaction between antibodies and specific antigens when they come closer to a distance of several nanometers. It is the binding of paratopes of antibodies to specific antigens or their epitopes that initiates a series of immunological responses to act against the respective antigens for their removal or destruction. So, reaction antigen plus antibody, this symbol represents a reversible reaction or equilibrium, so which gives antigen antibody complex that result into immune response. So according to the law of mass action, antigen antibody complex divided by antigen antibody equals to Ka by Kd equals to Keq. So this here Ka is the rate of the association constant, Kd is equals to rate of the dissociation constant, Keq is equal to equivalence constant. At equilibrium, the ratio of the concentration of product, that is Ag AB complex, and reactants, that is antigen antibody, are constant and called equivalence constant. What is an antigen, which is represented by Ag? So, anti means against, gen things that produces or causes. So, antigen, any foreign substances including immune response. So, any foreign substances that, when entering our body, sometimes self elicit a series of immune responses and are precisely called immunogens. Happens where some of them don't directly elicit an immune response but require the help of some other molecules to do so and are called haptens. Can be proteins, lipids, peptides, or polysaccharides. Epitope is antibody binding site. It is called as epitope. Abbreviation of antigen is Ag. What is an antibody? An antibody is simply the component produced by the immune system in response to antigen. So basically, antigens are the generator of antibodies. They interact with each other to induce an immune response. So glycoproteins produced by plasma B cells, also called immunoglobulins, it is Y-shaped structured. Antigen binding site is called paratope, and types of antigens are IgG, IgA, IgM, IgE, and IgD. What is an antigen antibody interaction? So, the interaction between antigen and antibodies are known as antigen antibody reaction. The reaction are highly specific, and an antigen reacts only with antibodies produced by itself or with closely related antigens. So recognition, antibodies recognize molecular shapes, that is epitopes on antigens. Generally, the better the fit of the epitope in terms of geometry or chemical character to the antibody combining site. So the more favorable the interaction that will be formed between the antibody and antigen, and the higher the affinity of the antibody for antigen. So the affinity of the antibody for the antigen is one of the most important factor in determining antibody efficacy in vivo. So antigen antibody interaction is a biomolecular irreversible association between antigen and antibody. The association between antigen and antibody includes various non-covalent bonds, intrinsic determinant, and variable region, that is VH by VL domain of an antibody. It is a diagrammatic representation of antigen and antibody interaction. So these diagram illustrates antigen antibody interaction. The central yellow structure is the antigen, which has specific regions called epitopes on both the side. The blue and pink structures are antibodies. Each antibody has a region via variable part that specifically binds to an epitope on the antigen like a lock and key. So the connection between the antigen epitope and the antibody forms the antigen antibody complex. This binding is crucial for the immune system to recognize and neutralize any foreign substances. So in summary, total, the diagram shows how antibodies attach to specific sites, that is epitope, on an antigen, forming complexes essential for immune defense. Properties of antigen-antibody interaction. It is highly specific reaction, occurs in observable manner, non-covalent interaction, that is van der Waals, ionic bonds, hydrogen bonds, hydrophobic interactions. So no destruction of antigen or antibody. It is a reversible reaction. Affinity, it is the strength with which one antigen binds on a single antigen binding site on an antibody. Avidity, it is a broader term that affinity, it is a measure of the overall strength of the antigen antibody complex. It depends on the affinity of the antibody valency of antibody and antigen, and the structural arrangement of epitopes and paratopes. Cross reactivity, it refers to the ability antibody to bind to similar epitopes of different antigens. Stages of antigen-antibody interaction. There are two stages of antigen-antibody interaction. First is primary stage, second is secondary stage. So primary stage, it is the initial interaction between antigen and antibodies. It is rapid, reversible, without any visible effects. Secondary stage, it is the irreversible interaction between antigen and antibodies. It is a slower process and with visible effects. Factors affecting antigen antibody interaction. So many factors affect antigen antibody reaction. So some of the common factors are: first is temperature. It depends on the chemical nature of epitopes, paratopes, and bonds involved. Example: hydrogen bonds are stable at low temperature and hydrophobic bonds are stable at high temperature. Second is pH. Optimal pH ranges is 6.5 to 8.5. So extreme pH values changes the conformation of antibodies and inhibit the reaction. Third is ionic strength. The effect of ionic is important in blood group serology. Here the reaction is significantly influenced by sodium and chlorine ions. So Na is sodium, Cl is chlorine. These ions in normal saline solution, Na plus and Cl minus cluster around the complex and partially neutralizes charges. So potentially interfering with antibody binding to antigen. This could be problematic when lower affinity antibodies are used. Fourth is enzyme treatment. So enzyme treatment, many proteolytic enzymes are used to enhance the antigen antibody reaction. Some of the commonly used ones are papain, which is present in papaya, and phycine bromelain. Fifth is concentration of antigen and antibody. Increase in the concentration of antigen and antibody enhances the reaction. Sixth is number of binding sites. Number of antigen binding site. So more the number of antigen binding sites on the antibody, the more the chances of interaction. For example, IgM is a pentameter and hence has 10 binding sites, whereas IgG is a monomer and hence has only 2 binding sites. So IgM will bind more efficiently with antigens. Structural arrangement: if the structure of the epitope and paratope is such that they could fit well as lock and key, then it enhances the interaction between antigen and antibody. I was talking about the structure, lock and key, epitope and paratope. So chemical bonds in antigen-antibody interaction. The interaction between the antibody binding site and the epitope involves exclusively non-covalent bond. In a similar manner to that in which proteins bind to their cellular receptors or enzymes bind to their substrates. The binding is reversible and can be prevented or distorted by high ionic strength or extreme pH. So the following intermolecular forces are involved in IgG and in antigen and antibody binding. So first we'll start with electrostatic bonds. These results from the attraction between oppositely charged ionic groups of two protein side chains. For example, an ionized amino group NH4 plus on lysine in the antibody and an ionized carboxyl group on an aspartate residue in the antigen. So hydrogen bonding, where the antigen and antibody are in very close proximity, relatively weak hydrogen bond can be formed between hydrophilic groups. Third is hydrophobic interaction. Hydrophobic groups such as the side chains of valine, leucine, and phenylalanine tend to associate due to van der Waals bonding and in an aqueous environment. So excluding water molecules from their surrounding, as a consequence, the distance between them decreases, enhancing the energies of attraction involved. This type of interaction is estimated to contribute up to 50% of the total strength of antigen and antibody bond. Fourth is van der Waals forces. So these forces depends on interaction between the electron clouds that surround the antigen and antibody molecules. The interaction has been compared to that which might exist between alternating dipoles in two molecules. So alternating in such a way that at any given moment, oppositely oriented dipoles will be present in closely opposed areas of the antigen and antibody molecules. Last is short distance, less than 1 Armstrong. So each of these non-covalent interaction operates over a very short distance, generally about less than 1 Armstrong. So antigen antibody interaction depends on a very close fit between antigen and antibody. So strength of antigen antibody interaction, first is affinity, second is avidity, third is cross reactivity. Types of antigen antibody reactions. So there are basically two types, in vivo and in vitro. In vivo, occurring in natural condition, it includes the general antibody mediated immune response occurring in our body against antigen. Second is in vitro, done in artificial condition. It includes a series of serological tests performed in laboratories to detect antigen or antibodies in case of many diseases. So in vivo, reactions include agglutination, precipitation, complement fixation, neutralization, ADCC, that is antibody dependent cell mediated toxicity, opsonization and immobilization. And in vitro, ELISA, RIA, Western blotting, immunofluorescence, ICT is immunochromatography, precipitation, and last is agglutination. So precipitation reaction, the reaction between soluble antigen and soluble antibodies forms an insoluble precipitate. The proportion of antigen and antibody in the reaction must be equivalent for the reaction to occur. It requires in the presence of electrolytes at specific pH and temperature. Antibodies involved are called precipitants. So types in solution are flocculation test and ring test. In agar, immunodiffusion. In agar with an electric field is immunoelectrophoresis. Agglutination reaction. So the reaction between insoluble large antigen and soluble antibodies leads to agglutination reaction. So formation of visible clumps is shown in agglutination reaction, occurs on the surface of the particle involved. Types are direct and passive. So direct is active, slides, tubes, heterophile agglutination test and Coombs is involved in direct test. In passive test, latex, hemagglutination and coagulation test is involved. ELISA enzyme-linked immunosorbent assay. It is one of the sensitive techniques for the detection of the presence of antigen or antibody and qualification as well in case of clinical diagnosis of many diseases such as AIDS, Ebola, parasitic infections, different parasites infections, and etc. Enzymes are used for labeling. So it detects free antigens or antibodies. Reagents include coating buffer, blocking buffer, wash buffer, substrate, samples, and assay diluents. Types are direct, indirect, and sandwich ELISA. Complement fixation and hemolysis. So complement fixation, it is the process of binding or fixing the serum complement proteins with the antigen antibody complex which further initiates a series of immune responses against the antigen. So it is used in the detection of any specific antigen or antibody in the patient's serum. Reagents include antigen, antibody, complement, and indicator system. Common complement fixation test is Weismann's test for detection of syphilis. Hemolysis, when the antigen antibody interaction results in the rupture of lysis of RBCs, it is called hemolysis, which results in the release of hemoglobin. It can be catalyzed by enzymes called hemolysins. It is the demonstrable endpoint of some reactions. Immunofluorescence and RIA. Immunofluorescence, it is a type of immunoassay technique in which fluorescent dyes are used for the visualization of antigen and antibody reaction. It detects surface antigens or antibodies. Fluorescent dyes such as fluorescein, isothiocyanide, and lissamine rhodamine are used. Types are direct and indirect. RIA, radioimmunoassay, RIA, it is a type of immunoassay in which radioisotopes are used for labeling the antigen or antibody to detect the formation of the AGAB, that is antigen antibody complex. So it can determine very small quantities of antigen and antibody in the serum. It is used for the quantification of hormones, drugs, and viral antigens. Applications of antigen antibody interactions. So, the most common use is the determination of blood groups, that is, blood typing, rapid diagnostic test kits used for pregnancy detection as well as detection of several diseases such as malaria, dengue, etc., are based on this principle. They require very little time for the test. Serological ascertainment of exposure to infectious agents, quantification of drugs, hormones, viral antigens, etc., to study the characteristics of different immunodeficiency diseases, to perform confirmatory tests for infections such as Western blotting for HIV infection. These are the applications of antigen and antibody interactions. Hypersensitivity reaction. Hypersensitivity is increased reactivity or increased sensitivity by the animal body to an antigen to which it has been previously exposed. The term hypersensitivity is often used as a synonym for allergy, which describes a state of altered reactivity to an antigen. Hypersensitivity has been divided into categories based upon whether it can be passively transferred by antibodies or by specifically immune lymphoid cell. The most widely adopted current classification is that of Coombs-Engel, which designates immunoglobulin-mediated, that is, immediate hypersensitivity reaction types 1, 2, and 3, and lymphoid cell-mediated hypersensitivity or cell-mediated immunity as a type 4 reaction. There are 4 types. First is allergic, second is cytotoxic, third is immune complex, fourth is delayed or cell-mediated. So hypersensitivity generally represents the dark side, signifying the undesirable aspects of an immune reaction, whereas the term immunity implies a desirable effect. A hypersensitive response, HR, is an antipathogen response in plants produced by AVR R system activation that leads to alteration in calcium Ca plus flux, MAPK activation, and NO and ROI formation. So there is a rapid necrosis of plant cells in contact with the pathogen. These process prevent spread of the pathogen and releases hydrolytic enzymes that facilitate injuries to the pathogen's structural integrity. Factors and causes of hypersensitivity. So causes of hypersensitivity, immune responses that are the cause of hypersensitivity diseases may be specific for antigens from different sources. So sources will be autoimmunity, reactions against self-antigen, microbial reactions, reactions against microbes, reactions against non-microbial and environmental antigens. Mediators will be cytokines, complement, mast cell, and eosinophils. Mechanisms of hypersensitivity. So hypersensitivity means exaggerated or inappropriate immune response which results into cell and tissue injury. Classification will be based on antibody dependent mechanism, T cell dependent mechanism. So many hypersensitivity diseases involve both humoral as well as cell mediated immunity. Immediate, which is type 1 hypersensitivity, it is caused by IgE antibodies specific for environmental antigen, and it is the most prevalent type of hypersensitivity disease. Immediate hypersensitivity diseases commonly grouped under allergy or atopy are often caused by activation of interleukins. So IL-4, IL-5 and IL-13 producing these allergies. So Th2 Th2 cells and the production of IgE antibodies which activate mast cell and eosinophils and induce inflammation. So mechanism will be allergen exposure which result to IgE production, that is mast cell activation, histamine release, last which leads to inflammation. Antibody mediated, which is type 2 hypersensitivity. IgG and IgM antibodies specific for cell surface or extracellular matrix antigen can cause tissue injury by activating the complement system, by recruiting inflammatory cells, and by interfering with normal functions. So outcome is tissue injury and cell destruction. Immune complex mediated, which is type 3 hypersensitivity. So IgG and IgM antibodies specific for soluble antigens in the blood form complexes with the antigen, and the immune complexes may be deposited in blood vessels in various tissues causing inflammation, which is no inflammation and thrombosis and tissue injury. So results is tissue injury and vascular damages. T cell mediated, that is type 4 hypersensitivity. In these disorders, tissue injury may be due to T lymphocyte that induce inflammation or directly kill target cell. So in most of these diseases, the major mechanism involves the activation of CD plus 4 helper T cells which secretes cytokines that promotes inflammation and activates leukocytes, mainly neutrophils and macrophages. So CTLs contribute to tissue injury in some cases. Types of hypersensitivity, that is 1 to 4. Type 1 IgE mediated, immediate. Example is asthma, anaphylaxis. Type 2 is IgG, IgM mediated cytotoxicity. Example is hemolytic anemia. Type 3 is immune complex deposition. Example is SLE. Arthros reaction. Type 4 is T cell mediated, delayed. Example is tuberculin test contact dermatitis. Types of hypersensitivity diagrammatic representation. So type 1 is IgE mediated hypersensitivity. You can see the example. Type 2 is IgG mediated cytotoxic hypersensitivity, antibody dependent cellular toxicity, and complement activation will be seen in type 2. Type 3, immune complex mediated hypersensitivity, and last is type 4, cell mediated hypersensitivity. So I'll give an overview of each type. Type 1 alternative name is allergic hypersensitivity. Type 2 alternative name is cytotoxic hypersensitivity. Type 3 is immune complex hypersensitivity, whereas type 4 is cell mediated delayed type of hypersensitivity. Principle of each types of hypersensitivity. Type 1 antibody mediated degranulation of granulocytes, which leads to destruction of cells. Type 2, antibody-mediated destruction of healthy cells. Type 3, antigen-antibody complex-mediated destruction of cells. And last, type 4, T lymphocyte-mediated destruction of cells. Primary mediator of all hypersensitivity, type 1 primary mediator is IgE, type 2 is IgG and IgM, type 3 IgG, IgM mediator, last type 4 CD plus for helper T cells or CD8 plus cytotoxic T cells. Other components of hypersensitivity, type 1 include mast cell, basophils, and histamine. Type 2 include complement neutrophils. Type 3 include complement phagocytes and K cells. Last, type 4 dendritic cells, macrophages, and cytokinin. Reaction type of hypersensitivity. For type 1, reaction type is mediated or after few hours. For type 2, it is approximately 5, almost 24 to 48 hours. Antigen and origin. So for type 1, antigen is soluble and its origin is exogenous. For type 2, antigen is fixed on cells and its origin is endogenous or exogenous. For types, either exogenous or endogenous. For type 4, it is soluble and cell-bound antigen and origin is either exogenous or endogenous. Antibody and mechanism. For type 1, antibody IgE is fixed on mast cell and basophil. Mechanism IgE cross-linking, which turns to mast cell degranulation. For type 2, IgG or IgM free in circulation. Its mechanism that leads to complement activation, and IgM free in circulation. Antigen antibody complexes, which leads to complement activation, and last, tissue damage. Type 4 hypersensitivity, it's antibody, it's not applicable, and the mechanism Th2 cells secrete cytokines, which leads to macrophage and CTL activation. Complement and appearance transfer. For type 1, complement activation is absent. There's no complement activation. Appearance is will and flare. Serum transfer, passive transfer is possible. For type 2, complement activation is present. Lysis and necrosis will happen and then passive transfer. Type 3, it is present complement activation. Appearance is erythema and edema. Edema is nothing but swelling. Serum is passive transfer. Type 4, complement activation is absent, no complement activation. Appearance is erythema and induration, and serum transfer only via T cells, not serum. Desensitization and example. So type 1 is easy and short-lived. Examples is asthma, rhinitis, eczema, and short desensitization. Example is Rh incompatibility, transfusion reaction, drug-induced hemolytic anemia. Type 3 is again easy and short-lived. Glomerulonephritis, lupus, vasculitis, farmer's lung. Type 4, difficult and long-lived. Its example is tuberculosis, granuloma, contact dermatitis, type 1 diabetes. So this is a diagrammatic representation of each type of hypersensitivity. Hypersensitivity type 1 immediate reaction. So some antigens are allergens such as insect venom, food, pollen, dust mite can induce the formation of IgE antibodies in individual with a corresponding. So predisposition, the IgE antibodies bind via Fc receptors to mast cells and sensitization, if the individual is re-exposed to the allergen, cross-linkage of the membrane-bound IgE occur. So this results in the immediate release of mediators. Example, histamine, which induce vasodilation, smooth muscle contraction, mucus secretion, edema, or skin blisters. So most allergens are small proteins that can easily diffuse through the skin or mucosa. They are frequently proteases and are active at very low dose. So IL-4 favors the differentiation of Th2 cells. The exact mechanism that leads B cells to produce IgE is not known. So the allergen stimulates the induction of CD plus 4 cells. These T cells secrete cytokines that causes IgE production by plasma cells. So the IgE molecule will bind to the Fc receptors on mast cells and basophils, which in turn causes vasodilation, increased vascular permeability, and vascular spasms. This type may occur as a systemic or local reaction. Second is hypersensitivity, type 2 antibody mediated cytotoxic reaction. Antigen during pregnancy is a typical example of type 2 reaction. Children who inherit the RHD erythrocyte antigen form from their father can induce immunization against RHD plus their RHD come in contact with the maternal RHD.
Hello. Namira madam.
Am I audible?
Hello.
Hello, sir.
Please tell madam.
Am I
No madam,
audible?
some voice is lagging actually.
Yes, sir. I guess network issue.
Net issue. That's what.
Mhm. So
I
this is
think
an
from
example of
hypersensitivity
type
to we
2.
have to. Yeah, yeah, yeah.
Yes, sir. Yes. This is antibody reaction. Hypersensitivity type 3 immune complex reaction. So
Madam,
in this antigen and immune complex,
madam,
hello,
madam,
yes sir,
will
yes
you
sir,
repeat hypersensitivity type 2?
type 2, yes sir.
Because actually net
Okay, so
issue.
this is type 2 network
Hmm.
issue. Yes, yes
Yeah.
sir. Yes sir, I'll explain. So, type 2 cytotoxic antibody reaction. So, here what happened? The immunization of individual to erythrocyte antigen during pregnancy is a typical example of type 2 reaction. Children who inherit the RHD erythrocyte antigen from their father can induce immunization against the RHD antigen in their mother. So, sensitization usually occurs at birth when fetal blood cells come in contact with maternal immune system. So in any subsequent pregnancies, maternal anti-RHD antibodies of the IgG type can pass into the placenta and cause severe hemolysis of fetal RHD erythrocyte. Other examples are drugs, example penicillin, can passively bind to erythrocytes Antibodies directed against penicillin then lead to lysis of erythrocytes. Lysis will happen here. So the formation of antibodies redirected against the basement membrane of the glomerulus can develop during the course of kidney inflammation. Lung damage accompanied by pulmonary hemorrhage and renal inflammation may occur due to cross reaction of these antibodies with the basement membrane of the lung. So in this type, antibodies are formed against the target antigen that are cell membrane components, not really hypersensitivity, but cytotoxic reaction. So this is an overall process for cytotoxic antibody reactions for type 2 cell-mediated, antibody-mediated cytotoxic reaction. Hypersensitivity type 3 immune complex mediated reaction. So here antibody antigen complexes can form during an immune response. Immune complexes can settle in vessel walls, the basement membrane of the lungs or kidneys, and in the joints. They can induce inflammatory processes in these structures by binding complement factors C, these C3A and C5A. So a particular type 3 reaction is the Arthros reaction when an antigen has penetrated the skin of an individual who has performed IgG antibodies. The immune complexes can bind to Fc receptors and most cells inducing degranulation inflammatory cells are recreated and complement is activated, which leads to the release of C5A and to local inflammation, platelet accumulation, and eventually to blood vessels, occlusions with necrosis. So this type is mediated by antigen antibody complexes which initiate an inflammatory reaction in the tissue. So there are two patterns of immune complex mediated injury. First is systemic and second is local. Type 4 hypersensitivity, that is delayed type hypersensitivity reaction. Haptens. These haptens are molecules of very small molecular weight. They are too small to function as antigens, but they can penetrate the epidermis and binds to certain proteins in the skin, dermis. Hapten carrier complexes are bound by antigen presenting cells of the skin, which then migrate to regional lymph nodes. T cell stimulation then occurs at the lymph node. The so-called sensitization phase lasts 10 to 14 days. So if the individual is re-exposed to the hapten, so they also cause edema formation or local inflammation of cytokines. Hello, am I audible?
No, no, some little bit sound is lacking actually.
Network issue is yes, sir. Yes, sir.
Yeah.
Yes, sir. So compounds containing and chemicals such as those found in this are typical triggers of type 4 hypersensitivity reactions. So actually there is two phase in this. First is acute and second is chronic. So in acute phase, you can give example of tuberculin test, contact dermatitis. In this mediated by CD4 plus helper T cell. Second is chronic. So granuloma formation mediated by CD8 plus cytotoxic T cell. You can give example of these. Conclusion. Antigen antibody interaction forms the foundation of immune response. It is critical for diagnosis, treatment, and understanding of hypersensitivity. The study aids in vaccine detection and therapy. Sorry, for question part. So there is 10 important questions for 3 marks. Define an antigen and antibody with example. What is the difference between epitope and paratope? I have shown lock and key structure for that. State any three factors affecting antigen and antibody interaction. Differentiate between affinity and avidity. Any 3 points you can mention. Name the two main stages of antigen and antibody interaction and mention one characteristics of each. List three chemical bonds involved in antigen and antibody interactions. What is cross reactivity in antigen and antibody reaction? Give two examples of in vivo and in vitro antigen antibody reaction. Define hypersensitivity and name the four types according to Gel and Coombs classification. In this, you can mention all four types: type 1, type 2, type 3, and type 4. Mention any three application of antigen and antibody interaction. For 5 mark questions, explain the law of mass action in the context of antigen antibody interaction with its equilibrium concept. In the beginning, I explained this law of mass along with this formula. You can mention it over here. Describe the differences between agglutination and precipitation reaction. You can give any reactions with example. You can give example of both the types at last with stage of antigen and antibody reactions. Both the stages I guess there is some net. Hello. Hello.
Yeah, yeah, please.
I
Please.
guess there's some network issues. Sorry for that.
No, no.
So I was explaining this 5 mark question. So differentiate between type 1 and type 2 hypersensitivity reaction with examples. Write a note on ELISA and its types and applications. Discuss the primary and secondary stages of antigen and antibody reactions. Explain
Madam, there is a serious network issue. Hello.
Describe the type pair in vastis. Write short notes.
Hello.
Hello. Yes, sir.
Madam, I think today's session,
Yes,
due
sir.
to internet problem, I think it is some serious
Yes,
concern.
sir. Yes, sir. Yes,
So we
sir.
can repeat once again. So we'll
Sure,
check.
sir.
We will check if this class is okay means okay. Otherwise, we can repeat once again. Because so many times, no, there is a due to
There's a
network
network
issue.
issue. Yes, sir. Yes, sir.
Whatever you have spoken, I think it won't be recorded. I think so.
All right, sir. No problem.
Yeah,
We'll
yeah,
continue.
please.
We'll repeat this class.
We'll see
All
tomorrow
right, sir.
in
All right, sir.
between.
No problem.
Yeah, ma'am.
Okay, sir.
So with
Thank you.
this, thank you so much, madam. You
Thank you,
have
sir.
nicely explained about antigen antibody
Yes,
and
sir.
as well as hypersensitivity reactions.
Yes, sir.
Hypersensitivity 1, 2, 3, 4,
Yeah.
and this is a dry subject actually in microbiology.
Okay, all right, sir.
So students should understand
Yes,
what is
sir.
the importance of antigen
Important.
antibody
Correct,
reaction.
sir. Lifetime it will
And
be useful for
yeah,
them.
yeah, yeah, that's the main thing. And as well as hypersensitivity reactions also.
Yes, sir.
So once
Allergen.
they understand the subject, no, so they
Yes,
can
sir.
write easily in
Easily in
exams
paper.
also. Yeah,
Yes, sir.
yeah.
All right.
Thank you so much. So
All
we'll
right.
see first, check the content. Whether
Yes,
it
sir.
is okay means no problem. So
Sure,
otherwise
sir.
we will repeat it. Okay.
No, no problem, sir. We
Yeah,
can
yeah.
do.
Thank you. Thank you,
Thank you,
ma'am.
sir. Thank you.